Good afternoon. Uh, First of all, before studying today's demo class, I would like to give a brief introduction about myself. Hello, my name is Arvindo De Burma, and I am from Tripura. I have completed my MTech in biotechnology from IIT Guwahati in the year 2020. I have also a brief work experience of 3 years. 2 year I have worked as a research associate in IIT Guwahati, and also I have an opportunity to go and work for 1 year at Gifu University, Japan as a research assistant. Apart from my research work, I had also been engaged as a teaching assistant during this research program. My major work was to prepare notes and take classes time to time to BTech and MTech student, and also help the BTech and MTech student in the lab to carry out their research work, preparing their reports, and also for their thesis preparation. So this is a bit brief introduction about myself, and I really love teaching. I think because of this teaching assistantship, I have also got my love or likeliness to become a teacher. And I strongly feel that as a research scholar, I have accumulated basic knowledge about life science, which I would like to share my knowledge with the student and make them understand their basic concept that will enable them to crack their exams, both national level and international level of exam. And yeah, this is why I have chosen to become a teacher, so that I can help the student, make them understand the basic concepts, especially in life science. So once again, I would like to say thank you and would like to welcome in my demo class today. I have chosen photosynthesis for my topic, since I have completed my degree in uh, biotechnology, and it's part of a life science. And photosynthesis is being an important topic in the life science. Why? Because we can say this is the like primary thing to make possible of survivability of a life in the art. Because without photosynthesis, it's not only about the plant, even the animal would not have able to survive if there is no photosynthesis. As we all know, that photosynthesis is a primary process through how the plants prepare their own food, so in presence of sunlight. As we say, photo means sun or light, and synthesis means preparation. So the photosynthesis means it is the preparation of food in presence of sunlight by the plants. Therefore, this plant that produce food by converting the light energy into chemical energy has played an important role for the survival of the living beings on this earth, because these plants are now considered as a primary source of food for all other living beings. And in turn, plant being an autotrophic organism, that is some organism that can produce their own food, is called as autotroph or autotrophic organism. And here the plant utilize sun as their source of energy to prepare their own food. Just like we can say, if you take an example, like in the if you want to prepare a food, we need gas, we need water, we need uh, rice, and then you prepare a food for yourself. Here, the plant is the one who is preparing their food, and you can say the gas can be considered as the sunlight that is being used during this process, and also the water and the rice. Here we can say carbon dioxide could be the water, sorry, could be the rice, and water is obviously we all need for it, and then you prepare a food that is in the form of glucose in case of plant. Therefore, the plant produce their food in presence of sunlight. That's why it is called as photosynthesis. Now, let us try to understand the definition given in the book. That means photosynthesis is a biological process by which plant, in presence of uh, sunlight and chlorophyll, they use the carbon dioxide molecule and water to prepare glucose and oxygen as well as ATP in the form of energy. Now, yeah, photosynthesis is a biological process by which the green plants, not all type of plants, but generally it is the green plants and certain other organisms such as the bacteria or the some algae or some sulfur bacteria. Those are some of those bacteria that can produce their own food in presence of sunlight and converted this light energy into chemical energy. Therefore, I have mentioned the equation that is carbon dioxide plus water in presence of sunlight and chlorophyll. They produce glucose and oxygen, also ATP. Now, if we go to the chemical equation, the photosynthesis chemical equation, to prepare one molecule of glucose, you need six molecule of uh, carbon dioxide, six molecule of water to prepare one molecule of glucose, and six molecule of oxygen is released during this process. Therefore, to create one molecule of glucose, you need around six molecule of carbon dioxide. Our this requirement and number of requirement of water, carbon dioxide, that we will also study in our further slides. Now, since we already know that photosynthesis is a process where you require multiple ingredients, we can say, like carbon dioxide, water, sunlight, chlorophyll, etc. So, how this has been proved, how these elements are being used during this photosynthesis process, there our scientists have done various type of experiment in order to prove that their sunlight is required, carbon dioxide is required, water is required, chlorophyll is required. There, with the small experiment, they have find out all the elements, how they take part in the process of photosynthesis. So let's start with one by one, that this experiment that scientists have carried out. First experiment that has been carried out is called the variegated leaf experiment. What exactly in this experiment was done is, they have taken the leaf, and a part of the leaf they have closed it with a tap or some cover it so that sunlight cannot fall in that part of the leaf, and other part of the leaf were exposed to the sunlight. Now, if there is a sunlight, as we know, sunlight plays an important role in photosynthesis. That means photosynthesis will occur and glucose will be produced. Now, during the photosynthesis, the glucose that produced in the plant were not kept in the form of glucose, but number of molecules of glucose are joined together. Hundreds to thousands of glucose are joined together, and they are stored in the form of starch in the leaf or in various part of the plant, especially in the vacuoles of the cells. Those stars will be stored. Now, since I think you all of have done your chemistry experiment, where you have seen, if in the stars test, if you add a few drops of iodine, this stars is converted into blue. So this same principle is applied here in this experiment also. Now, those leaf that has been exposed to the sunlight, this particular region where it is in a form of green, when you put iodine over there, this area it turns into blue, because it means this area has produced or like has carried out photosynthesis, and stars has been stored here. Therefore, when you add iodine, this particular area has turned into blue. On the other hand, in the white region, which you can see where there is no sunlight, that area, that means there is no photosynthesis occur, and when you add iodine, there is no blue color formation, because there is no storage of stars was there. Therefore, that region become white. This experiment thus proved that sunlight play an important role for the formation of stars through photosynthesis. Next, in order to prove the presence or the requirement of carbon dioxide as a part of important part or elements during photosynthesis, the potassium hydroxide soaked cotton experiment, or the KOH soaked cotton experiment, was conducted. This particular experiment was done by a scientist called Molly. Therefore, this experiment is also called as Molly's half-leaf experiment. In this experiment, what m- uh, the scientist Molly has done is he had taken a jar with a cotton and KOH over there, and put the half of the leaf inside the jar and tightly close it, and another part of the leaf was exposed outside. Now, whatever inside it is there, if there is any presence of carbon dioxide, if even if the plants also reduce any carbon dioxide, whatever, uh, all that excess carbon dioxide in this region will be like taken out by the presence of potassium hydroxide. And on the other hand, in the outside, the leaf will be totally exposed to the atmospheric carbon dioxide. Therefore, inside, whatever carbon dioxide is present in the bottle, everything has been soaked up by the potassium hydroxide. Now, once it was exposed to the sun, and when it is taken out, the leaf inside the bottle where KOH is present, when you add iodine in it, again there is no blue color formation, but to those leaf that has been exposed outside, there was a blue color formation, indicating CO2 was used for the purpose of photosynthesis. And in case of the uh, in case of the leaf that has been kept inside the bottle, no photosynthesis occur over there, because there was no CO2. Therefore, the blue color was formed only in the upper part of the leaf, not in the down. Another scientist carry out one more experiment called the bear jar experiment. In this, he just want to know like the important elements like carbon dioxide, oxygen, and the light plays an important role in the process of photosynthesis. Although during that time, still the scientists couldn't tell about the presence or the release of oxygen during the process of photosynthesis, but yet they consider that certain amount of uh, gases like carbon dioxide, oxygen, and light is important for the source. So in this experiment, since we know plant need light for the production of photosynthesis, and it also need oxygen to burn, that is the major reason, because during these experiments scientists didn't know that oxygen is also being produced during photosynthesis, but they know one thing is like the fire to burn it needs oxygen, and also the animal which is there, it also need oxygen for respiration to survive. So what scientists did is, when they put a mouse and a candle inside a jar and totally cover it, and no gas from the outside was allowed to enter inside the bell jar, after some time both the mice and the candle got suffocated, the mice got suffocated and died, and the candle also got extinguished. But when they put mint plant inside the bell jar, they were found like both the animal, the plant, and the light could survive. Now, in order to prove that the oxygen is being produced during this process, there is another scientist called Jan Ingenhouser experiment where what he did is he brought the aquatic plant. So friends, now I think you also know very well this aquatic plant, they can live inside the water, and in presence of sunlight they also produce or they also carry out photosynthesis, same like their counterpart that is in the terrestrial plant. They also can produce the photosynthesis. So what he did, he took a jar, put the plant, and when it is kept in front of the sunlight, the plant carry out the photosynthesis, and during this process oxygen being released is seen in the form of bubbles. On the other hand, when the bottle is kept, a bottle, water and the plant is kept far away from the light, there was no bubble formation. Hence, prove that there was no oxygen formation because there was no photosynthesis being carried out inside the bottle. The plant used for this purpose was hydrilla, and the plant produced oxygen during photosynthesis was proved by this particular experiment. Another scientist called Van Sek one in his one-sake experiment 1854, he confirmed that the glucose that is produced during photosynthesis is stored in the form of starch, and also the chlorophyll present uh, in the plant were located in a place called the chloroplast, or in an organelle called chloroplast, and the glucose are made only by the green parts of the plant. Now, as we have already mentioned in our definition that photosynthesis is carried out by the green plant in presence of sunlight, chlorophyll, and that convert carbon dioxide and water into glucose and oxygen. So that means through this experiment it is proved that only green part of the plant is produced, is where the photosynthesis takes place, because we know the plant has flower which may be of different color, leaves of different color, their bark is different color, roots are different color, trunks are different color, stems might be different color, but most of the plants will have a green leaf. That means only in this green part of the plant where photosynthesis is being carried out. Next, another scientist called Van Niel experiment. In this experiment, he also tried to prove out that the oxygen produced during photosynthesis is not from the carbon dioxide but from that of the water. So we know the carbon dioxide also have two molecules of oxygen in it, and water has a single molecule of oxygen in it. So this particular in during the time of photosynthesis, that means the oxygen in the carbon dioxide is not splitted to make the oxygen, but it was the oxygen present in the water that has been splitted from hydrogen, and the oxygen is being released. So in order to prove this experiment, he used like bacteria, purple bacteria. This bacteria doesn't use water and carbon dioxide to prepare their glucose molecule, but instead they use hydrogen sulfide, because this blue purple bacteria, they are mostly present in a very harsh condition like in the volcano area and all, where sulfur is there, where since it is so hot you won't get any water over there, but the sulfurs are very huge amount present over there in the form of hydrogen sulfide. This bacteria, when they use carbon dioxide and hydrogen sulfide to prepare their food, the sulfur is being released during this process, and hence he concluded that the oxygen that produced during photosynthesis is from the water, not from that of the carbon dioxide. And he also proposed a formula where CO2 plus two molecule of hydrogen with A, that can be either oxygen or that can be sulfur, produce glucose along with water and two molecule of A, which can be oxygen or sulfur. So in order to further prove this particular experiment that the oxygen is produced from the hydrogen, they also carried out certain other set of experiment. So I would just like to ask, like, how do we confirm that whether this oxygen is either from the carbon dioxide or from the hydrogen or from the water? How can we confirm that? A simple process, I think even in the chemistry, we have also studied about the radioisotope. So this radioisotope level, oxygens are being used during this process. So they have radio level, the oxygen of carbon dioxide, and when they see in the experiment, they have radio level the oxygen presence in the uh, so in their first experiment, what they have done is they have radio level the oxygen of the water, and they keep the oxygen of carbon dioxide as it is. And when after the photosynthesis is being carried out, when they study the presence of oxygen in the atmosphere, it was found that the oxygen has a radio level of radio level with isotope 18. And on the other hand, when in the experiment too, when they carried out and when they have radio level the oxygen of carbon dioxide, but this time the oxygen of water has been without any radio level. And when they check the oxygen produced after photosynthesis, it was without any of radioisotope level. Hence, it proves that the oxygen that we get during photosynthesis is splitted out from water molecule, not from that of the carbon dioxide. I think this is very clear. The experiment that has so far conducted to check whether which molecules or elements are required during photosynthesis and whether the oxygen is produced from the water molecule or the carbon dioxide during the photosynthesis. So in the next experiment, which was carried out by the Angelman experiment. It's also called Angelman, and it's also called as Angelman experiment. So here, Angelman, during his experiment, he wanted to know like which particular light rays is majorly responsible, or wavelength of light is majorly responsible for the photosynthesis. And through this process, he also find out that the chlorophyll or the pigment presence in the plants are responsible for photosynthesis. So as we have studied that the plant have pigment which is mostly green, but apart from the green pigment they also have certain other pigment such as it can be a red, which are almost the carotenoid. Within the chlorophyll also there is chlorophyll A and chlorophyll B, and also some leaves such as xanthophyll, etc. So in this experiment, what Engelmann exactly done is he had splitted the sunlight into the rainbow color or the vigor color, where in exposing in the prism it can be separated into red, orange, yellow, green, blue, purple, etc. And when he used some bacteria, autotrophic or photosynthetic bacteria, he found that most of this plant and he used this to check this plant, that is a Calabdophora plant, for the production of photosynthesis. And during the photosynthesis, since O2 will also, oxygen will also be produced, to check that he also used a group of bacteria, aerobic bacteria. So this aerobic bacteria will only go to the place where there is more number of oxygen, and more number of oxygen will be produced only when there is more photosynthetic activity has been occurred. So according to this experiment, what Engelmann found is like most of this bacteria were seen accumulated in the region that is a red or near to red, and blue or near to blue color. Hence he considered that this red and blue light are the most like responsible web length where photosynthesis occur. He has plotted a chart over here, which you can see that chlorophyll A, which is like 450 around, and another is like nearly 680 something around. You can see the peak of chlorophyll A with dark green color, and in the light green color, that is chlorophyll B, which is around might be 450 or 460, and some carotenoid in a orange in color. And in the B, you can see the rate of photosynthesis is higher, which coincide with that of chlorophyll A, which is around 430 to 450 nanometer. And in the other side, here also where we can see the rate higher rate of photosynthesis, which again coincide with that of the chlorophyll A, which is around might be 680 nanometer. With this, Angelman has proved that the rate of photosynthesis mainly occur in this region where it is 460 to 680 nanometer, and this is also region where you can find the chlorophyll A. Therefore, he concluded chlorophyll A is the main place where photosynthesis exactly occur, or it is the major pigment where photosynthesis takes place. On the other hand, chlorophyll B also, it is in this region also we can see some chlorophyll B, and also in the keratin region we can also see some rate of photosynthesis, but they are lower in compared to that of the region where chlorophyll A is found. Therefore, this chlorophyll B and carotenoids also carried out photosynthesis, but the majority of the glucose production or the photosynthetic activities takes place in chlorophyll A. Now, you may also ask then, like, what is the function of this carotenoid, chlorophyll B, or xanthophyll? Now we know there are there are few other plants. They are not green in color, they are red in color, they might be yellow in color. So these particular plants, they doesn't have chlorophyll A in them. In that case, carotenoid is the one, or pigments where most of this photosynthetic activities takes place. So where chlorophyll A doesn't work, there this chlorophyll B or carotenoid or xanthophyll will work, especially to the non-green plant. Another important function of this pigments will be like when there is a higher sunlight, and the higher number of other rays might be more yellow, more green, more orange apart from the red in color. During that time, the chlorophyll A alone will not able to take all this color, because it will use only either red and blue, but rest of the color it won't be using. But this color or this light intensity might damage the pigment. I think you can understand, because the like more amount. Just take an example. If you pass a water, very heavy speed of water towards a machine, the machine will get damaged, because if the water also contains some consider the water is not a pure, it has some impurities in it, dust is there, some floating materials are there, everything it goes together and put inside the machine will get damaged. So if you make other channel, so that you take carry out the floating material separately, you separate the sand separately, you separate the dust separately, and only a good pure water form goes to the machine will last longer. Similar way, if there is like higher sunlight, because we know the quality of sunlight may not be same every day. Sometimes there will be so high sunlight that chlorophyll A will get damaged because of this. During this process also, the other carotenoid will absorb all the light that chlorophyll A will not require. In this way, they prevent chlorophyll A from damage. So these are some of the function of this pigmented plant. Now, now as we study that chlorophyll is present in a specialized organelle of plant called as chloroplast. So let's try to study what is this chloroplast and what exactly is inside them. So the chloroplasts are made up of two membrane called inner membrane and outer membrane. In between, it has an intermembrane space. Inside them, there is a presence of thylakoid, or each thylakoid together form a granum. So you can just say like number of coins are put together and they form the granum. And they form the granum. Inside the thylakoid, it is also again made up of two membrane, and inside them it is called the lumen. So it is in the thylakoid membrane where the photosynthesis exactly occur, because in this membrane there is a presence of photosystem. And in case of plant, especially in the thylakoid membrane, there is two photosystem called photosystem 1 and photosystem 2. The grana are connected by the lamella, and this lamella also have also where photosynthesis occur, and here only the photosystem 1 is present. So, what we can find in this structure of chloroplast is right reaction occurs in the grana. The pigment system is present in the thylakoid membrane, and the dark reaction mainly occur in the stroma. Dark reaction occurs with the help of ATP and NADH produced during the time of light reaction. So, in our next slide, we will also study about how ATP and NADPH are produced during the process of light reaction. Each and everything we will go through in our next slide. Next, when we talk about the photosystem 1 and 2, where it is present mainly where are they present? In the membrane of the thylakoid. The dark green area is where the photosystem 1 and 2 are present. The photosystem 1, it absorbs at 700 nanometer. It absorbs the light at have 700 nanometer, and photosystem 2, it absorbs light at 680 nanometer. These are all part of the chlorophyll A. Now, the photosystem 1 and a photosystem 2 doesn't mean that they are like a serial manner. It is not in the form of photosystem 1 and photosystem 2 serially, but it is based on the discoveries they have been done by the scientists. So before they study before they know about photosystem 2, they have already studied photosystem 1, where they know 700 nanometer of light being observed, and this light electrons are being produced, and they help in photosynthesis process. Later the scientists also discover, like before photosystem 1, there is another system that also absorb light at 600 nanometer, and which is exactly the first acceptor of light and electron, and from this system it was passed to photosystem 1, and this system they have named as photosystem 2, since they have discovered this mechanism of the photosystem 1. So remember, it is photosystem 2 where first light is absorbed, electrons are absorbed, they are excited, and from photosystem 2 to they are transferred to photosystem 1, and from photosystem 1 finally they are sent or they are given back to NADP for the production of NADPH. So each and every of this step we will go through in our next slide. So, now since we started understanding about photosystem 1 and photosystem 2, may I know where they are? They are mainly present in the membrane of the thylakoid, is where both the photosystem 1 and photosystem 2 occur. Now, in our in our light reaction, where light is the main important part for the photosynthesis, in the light reaction we have two. One is called the non-cyclic photophosphorylation, and the other is called as cyclic photophosphorylation. So what exactly happened in a non-cyclic photophosphorylation? So water is splitted in presence of zinc and magnesium, and it is splitted into two electrons and two protons. This electrons is now accepted in the photosystem 2. After splitting of the water, they're accepted in the photosystem 2, where light is also absorbed at 680 nanometer. With the presence of this light, the electrons are being excited. What exactly mean by excited means the electrons now gain a power. Because of this power, the electrons are excited, and now they can move from one system to another system. During this, the electrons move from various cytochrome, like cytochrome Q, cytochrome PQ, cytochrome B, cytochrome F, etc., and plastic QAnon, and finally they are transported to photosystem 1. The electrons are transported to photosystem 1 after traveling to various cytochromes. Now, during this process of transport of electrons, there are also another coenzymes or elements that helps them to transport. These are copper and iron that helps in the transport of electrons to from photosystem 2 to photosystem 1. Now, during this process of transfer of electrons, ATP is produced from ADP. Now, this once the electrons are being transported to photosystem 1, again they lost their energy. So to make them in again in energetic state or in excited state, again at photosystem 1, light is absorbed at 700 nanometer, and these electrons are being further excited, and then finally by the ferredoxin reductase, the NADP accepts the electrons with two proton molecule and finally form into NADPH. So I think now you have understand how these electrons are being splitted from the water in presence of zinc and magnesium. They are splitted into two electron molecule and two proton molecule, and oxygen is finally separated. Remember, during this process, two molecules of water is required. Only one of the water is splitted and another water is given back. And the process mainly occur in the lumen. I think you also know which one is the part of the lumen. It is this light green part is called the lumen. That is, the water splitting takes place inside the thylakoid, and the like photosystems are present in the membrane of the thylakoid, and the transfer of electrons occur within the membrane of the thylakoids. So the once the photosystem to accept the electrons and excited by light at when it observed at 680 nanometer, this excited electron transported from one particular chamber to another, that is from different cytochromes. They are being transferred. During this transport of electrons, ATP is converted into ATP in the form of energy, and finally electrons are brought to photosystem 1. When electrons reach to photosystem 1, they lost their energy, and to regain their energy again, the S1 has received sunlight at 700 nanometer. During this process, again the electrons are excited, and they are further again moves to different cytochrome chamber, and finally the electrons are accepted by NADP plus two proton molecule and finally form NADPH2. Now, these electrons that was there, which has been splitted from the water and from photosystem 2, it took its journey and reached to photosystem 1, and finally it is accepted by NADP, and the electron doesn't come back to either photosystem 1 or photosystem 2. That's why this is called as non-cyclic phosphorylation, because the electrons doesn't come back to its original place or from where it has been generated. That's why it is called non-cyclic phosphorylation. Sometimes it is also called the jet model, because if you see them, it exactly look like a jet form. That's why it is also sometimes called as a jet model. Next, the cyclic photophosphorylation. In the cyclic photophosphorylation mainly occurs at photosystem 1. Now, that means since it occurs only in photosystem 1, means we can assume that this particular reaction mainly occurs at lamella, because in lamella, it has only one photosystem 1, and it doesn't have photosystem 2. Therefore, the cyclic reaction mostly occur in the lamella region. Now, during this process, again in the photosystem 1, when light is been absorb, the electrons get excited, reach to and this electron transported to different chamber, and again finally after producing ATP it comes back into chlorophyll. No NADPH is produced during this process because it doesn't give its electron to NADPH, but this electron come back to again photosystem 1. So it started from photosystem 1 after various part it has been traveled to various chamber of cytochrome, and finally it reached to photosystem 1 again. That's why this particular process is called as cyclic photophosphorylation. I think both non-cyclic phosphorylation and cyclic photophosphorylation has been understandable, and there is no doubt regarding this particular two process. So let's move with our next slide. So in our next slide, we will try to understand how this ATP is produced during the electron transfer. So, as we know that during the time of the splitting of water, hydrolysis, which we call, during that time the proton is produced, H plus is produced. This H plus, as I have already mentioned, the splitting of water mostly occur within the lumen, that is inside the thylakoid. The protons are now present in a very higher number in the thylakoid itself, inside the thylakoid itself. This lead to a electrochemical potential gradient. How? Because more number of protons are produced inside and less number of protons are present outside the thylakoid membrane. Not only that, this hydrogen like when there is a transport of electrons, as I have told you, there are certain molecule like the irons or copper which help the electrons to transport from one chamber to another, that is from one cytochrome to another type of cytochrome. During this process, these enzymes they may lose their structure. Now as I have all as you know, in our chemistry we have studied enzymes are the molecule or protein molecule that helps in a reaction which which make the reaction faster and complete the reaction very quickly, but during this process they doesn't change their structure. They will complete the work and they will leave back. It will be just something like a screwdriver where you use the screwdriver to fix the screw. Once your work is done, the screwdriver goes. There is no damage of the screwdriver. Same, our enzymes are also like that. They perform a particular reaction, complete the reaction and goes back. During this process, they neither gain, they don't gain anything, neither they lose anything. They will be in their same structure. But what exactly happened during the transport of electrons? Sometimes this enzyme may lose their balance because of carrying the electrons. So in order to say like, no, I don't want to make my structure imbalance, give me a proton so that it will be easier for me to carry the electrons from one chamber to another chamber. So during this process, the enzymes they again use the proton molecules which is present outside of the thylakoid membrane, inside the stroma, which is present inside the stroma. They will only use those hydrogen molecule, not from inside the thylakoid membrane. They will not take any thylakoid membrane from the they will not take any proton from the lumen, but they will be taking the they will be taking the proton from the outside of the thylakoid. Therefore, more number of hydrogen protons are being decreased in the uh, outside of the thylakoid membrane. And once this proton is being used, once the electrons are transferred to photosystem 1, the electrons are or not the protons are not released outside, but they are again released inside. This is the one that you can see they are released inside. Therefore, during water split also you can get proton, and during the transfer of electron process also you transfer the proton from outside into inside. This is how again you gain your proton inside and how you lost your proton outside. And also, as we told in our reaction, the final acceptor of electron is NADP, which is then converted into NADPH. This again use another hydrogen proton to get converted into NADPH. Therefore, more number of hydrogen proton is getting reduced outside the thylakoid membrane, and whereas inside it is increasing more and more. This leads to a very high electrochemical potential gradient, or the inside it is more higher proton and outside it is very less proton. And we always know that always a molecule will try to move from inside to, sorry, from higher concentration to lower concentration. Since here the inside you have more number of proton and outside it has very less number of proton, this hydrogen wants to come out inside. And during this process, they since we know our membrane doesn't allow any of these electrons or protons to easily pass through it. Okay, it needs certain bypass channel. So here the bypass channel CF1, there is cytochrome 1 F1, this is F naught. This cytochrome F naught is the one that helps in the transport of this hydrogen proton outside, including with CF1. So this diagram has a bit correction. Here it will be F naught, and the outside one, which is also called as ATP synthesis, it has F1. So this will help the hydrogen proton to come outside. But when you have more number of hydrogen proton outside, there will be more force or more pressure to come outside. During this process, there is like electromotive force generate. Due to generation of this electromotive force, ADP plus PI will form into ATP, and thus ATP is generated. So this moment is mainly osmotic pressure moment, and we know osmotic definition is the movement of molecules or chemical from higher concentration towards a lower concentration. So if there is a higher number of molecule inside and very less number of molecule outside, the movement will be more faster. So therefore this faster movement creates a electromotive force, and due to this it leads to the production of ATP. So I hope you can now understand how ATP is produced. So we already talked about how NADPH is produced. It accepts the final electron. NADP accepts the final electron, and in presence of two proton, it is converted into NADPH. And ATP is produced because of the electrochemical potential gradients of the proton presence inside the lumen, and when it comes outside, it creates an electromotive force, and due to this electromotive force formation, the ADP and PI forms into ATP, or adenosine triphosphate. Next, we will go through the dark reaction. Now, the dark light reaction I have talked about, it mainly occurs in the thylakoid membrane and in the lamella, where both cyclic and non-cyclic reaction takes place. Now, in case of dark reaction, it mainly occurs in the stroma. So, the stroma is the region or the gap region. So, this particular, all this white like area, this is your stroma where the dark reaction occurs. Now, the dark reaction doesn't mean it occurs in the light, or sorry, in the night. Dark reaction means when there is a no light or when there is a very less light, even during that time dark reaction can occur. So dark reaction doesn't mean that it only occur in the night, but dark reaction means the reaction of photosynthesis or reaction of glucose preparation that occurs in absence of sunlight or in a very less light. That condition is called as dark reaction. Now, dark reaction occurs actually in plants like C3 and C4 plants. So what is this exactly C3 plant or C4 plant? Let's try to figure it out. So since we know, okay, since we know that the carbon dioxide is the major component for the production of like glucose, so carbon dioxide is fixed by a enzyme called ribulose bisphosphatase. This is an enzyme where it is CO2 is combined with a molecule called ribulose phosphate. So ribulose based phosphate, it's a 5 carbon atom molecule where carbon dioxide is fixed or come and join and form 6 carbon molecule, a process called as carbon fixation. So once 6 carbon molecule is formed, they are broken down into 3 carbon molecule called as 3 phosphoglyceric acid. In the structure you can see now, from after C joined with ribosquid form the 6 carbon molecule, they are now broken down and form into 3 carbon molecule into 2 molecule of PGA, phosphoglyceric acid. And this phosphoglyceric acid, so when the carbon is fixed with ribulose base phosphate in presence of rubisco, the carbon is fixed and they are broken down into two molecule of compound, two molecule compound C3, two molecule C3 compound called phosphoglyceric acid. Now phosphoglyceric acid using two molecule of ATP, we will be studying the requirement of ATPs and NADPs. So now the phosphoglyceric acid using ATP, it is then converted into glyceraldehyde 3 phosphate into two molecule of glyceraldehyde 3 phosphate, and one of this glyceryl and during this process it will release out one carbon molecule, and then again it will convert it back into ribulose bis phosphate, which is again 5 carbon molecule in using ATP, and this particular process is called as regeneration of ribulose which have a 5 carbon sugar. So now, during this particular process, only one carbon will be released, which then further form into glucose. Now, in this figure, you can say one molecule of G, like glucose, glucose 3, glyceraldehyde 3 phosphate. This is because in the beginning we have already used 3 molecule of carbon dioxide. Therefore, when we say glyceraldehyde 3 phosphate, it has 3 molecule of carbon compound. So to make one glucose, we need 6 molecule of carbon. So if one molecule of carbon require one molecule of carbon require 3 molecule of ATP, because here 2 ATP is required, here 1 ATP is required. So together 3 ATP, and here 2 NADPH is required. So to fix one carbon, you require 3 ATP and 2 NADPH. Therefore, when you use 3 carbon, you require 6 ATP, because 3 into uh, 3 into 3, here it will be to make 3 carbon. Here again 2 molecule of ATP is required, 6 ATP. Here 2 molecule of NADPS is required, so 6 NADPS. Here 1 molecule of ATP is required, so it become 3 ATP. So totally, to make one glucose molecule, you need 6 carbon dioxide, 18 ATP, 12 NADPS. So how can you make this calculation? Is like you use 1 carbon dioxide require 3 ATP and 2 NADPs. Now, if somebody says 1000 carbon dioxide, to fix 1000 carbon dioxide, you will require 3 into 1000 ATP and 1000 into 2 NADPH. Through this way, the numerous number of carbon or glucose, you will able to find the amount of ATP and NADPH required for this process. Therefore, this process, no ATP is generated, but to produce glucose, you are using ATP and NADPH, right? Now, what happened? Ribosco is an enzyme which is also considered to be the highest number of enzyme present in the biosphere, because we already know the plants are more there. Every green plants have chlorophyll in them, so every chlorophyll will have Ribosco enzyme with them. Therefore, they are the highest number of enzyme present in our biosphere. Now, when there is a higher number of oxygen in compared to that of carbon dioxide, Ribosco, this enzyme has a tendency to bind with both oxygen as well as carbon dioxide. But only when higher number of oxygen is present, Ribosco will bind with oxygen. During this process, the plant, instead of C3 process, it will go a process called C2 process, where rubusco will accept carbon oxygen, and this oxygen will be now, with presence of rubusco, it will be converted into phosphoglyceraldehyde and phosphoglyceric acid, and this phosphoglyceraldehyde is again converted into phosphoglyceric acid. During this process, it needs ATP, and also during the fixation and from this phosphoglyceric acid to revolute bisphosphate process, again another ATP is used. Therefore, in this process, two molecule of ATP is used and a carbon dioxide is generated while oxygen is while oxygen is being taken and carbon dioxide is being produced. So here not only you lose ATP, but the plant also take oxygen and it also release the carbon dioxide. A process called photorespiration, because we know in the respiration process it is CO2 that is released, and in photosynthesis process it is oxygen is released. Here, since CO2 has been released during this process, this is called as photorespiration. There are some uh, like scientific telling, like you should not sleep in the night in the uh, under the tree, is because of this photorespiration process, because during that process the plant may take oxygen and it may release more carbon dioxide, leading to scarcity of oxygen within the environment, and the person who is sleeping below the tree might die because of lack of oxygen. So there is that time the plant may take oxygen. Now what is the benefit of this particular photorespiration? Is there is no benefit at all, because no glucose is produced here. You can see in the cycle only CO2 is released, but no carbon dioxide, no glucose is produced here, and two ATPs are being used. So only energy is being wasted, no glucose is produced. Therefore, this will leads to a loss of reaction in the plant, unlike C3 plant. Next, is about C4 plant. Now, since we know that C4, C3 plant and C4 plant are of different types. C4 plants are the plants that can be available in a very extreme condition, like uh, in the desert area, like the cactus and all, where if there is more amount of sunlight, if they started opening their leaf stomata, I mean, they will lose more number of carbon dioxide, water during this process, and the plant will die, because generally they have very less water in their surrounding, and if they open their stomata for carbon dioxide each and every time, that means during this process they will not only take carbon dioxide, but also they will lose more number of water, and the plant will not able to survive. In order to prevent this particular process, the C4 plant has adapted to a mechanism there where they don't need to like take carbon dioxide all the time. What they will do is, like, they will take carbon dioxide, keep them inside, and when they will need for the production of glucose, they will utilize this carbon dioxide for the production of glucose. So what we have seen in our previous C3 plant is this particular plant, this Calvin cycle or the glucose generation cycle, it C3 cycle mainly occurs in the mesophyll cells. You can see in the diagram the mesophyll cells are present in the leaf. These are the loose cell. They are not bundled. They are in the form of a loosely packed cell. Whereas there is another group of called the bundle sheets. These bundle sheets are a group of cells that are attached together firmly in a form of bunch. That's why they are called as a bundle sheet cells, and they are present in the C4 plant. Now, using the mesophyll cell, in C3, it will be the mesophyll cell where this CO2 fixation occur. In case of bundle cells, in case of C4 plant, the mesophyll cell will take the CO2 from the atmosphere, and in presence of phosphoenol pyruvate, the oxygen carbon dioxide will fix to the phosphoenol pyruvate and converted into C4 acid, which can be which are oxaloacetate and malate. So either in a form of oxaloacetate and malate, it will fix the C3 carbon and turn them into C4 carbon. The carbon dioxide is fixed with phosphoenol pyruvate and they are converted into oxaloacetate and malate. They are now transferred to they are now transported to the bundle sheet where this CO2 is again broken down and they are now converted back into a Calvin cycle. This CO2 is now used in the Calvin cycle. The C3 or the pyruvate is then again sent back to a mesophyll cell, regenerated into phosphoenol pyruvate. So this is how the glucose is produced in case of C4 plant. That is, CO2 from the atmosphere is taken by the mesophyll cell, where CO2 is fixed with phosphoenol pyruvate and converted into C4, that is oxaloacetate and malate, which is then transported to the bundle sheet. There, in the decarboxylation process, carbon dioxide is broken down, and this carbon dioxide is now used by Calvin cycle to produce glucose, and the C3 is then sent back to mesophyll cell where it is regenerated and converted back into the phosphoenol pyruvate. In this process, to fix one carbon dioxide, we need 5 ATP molecule and 2 NADPH, because we already know in the Calvin cycle we'll require 3 molecule of ATP and 2 molecule of NADPH. Here, during the carbon carboxylation process, another 2 ATP, extra 2 ATP is required. Therefore, totally in this process, 5 ATP and 2 NADPH is used. That means to produce one glucose, 6 ribulose bisphosphate, 6 molecule of carbon dioxide and 30 molecule of ATP and 12 molecule of NADPH is used during this process. That means compared to N3, C4 needs more energy in order to produce the glucose in the process. Now let's study some of the factors affecting the photosynthesis. What are the factors affecting photosynthesis? It can be both internal and external photosynthesis. What could be the external photosynthesis are like light, water, carbon dioxide, temperature and minerals. So we know light is required, water is required, carbon dioxide is required for the photosynthesis from our experiment. But the temperature can also play an important role because certain reactions takes place only in an optimum temperature, and the optimum temperature in a living cells can be 30 to 27 degree, where our enzymatic activities are higher. Therefore, low temperature and higher temperature may reduce affect the photosynthesis. Similarly, the minerals. So we have already studied copper, zinc, magnesium and iron, their role during photosynthesis. So if these elements are not there, then properly the reaction may not occur, and this might hamper the photosynthesis process. Now, what are the internal factors? Internal factors can be the pigment of the leaf. Young leaves will be more photosynthetically active because they are more green, because we know only the green part of the plant, that is chlorophyll A, has the highest rate of photosynthesis. So when the leaf become yellow, they may get reduced to photosynthesis production. Similarly, any plant that might be of pigmented plants, rate of photosynthesis in them will be less, because we know the activity of photosynthesis is more in the green region. Another is the leaf age of the leaf. As I have already told, young leaves will produce more photosynthesis because they are more green in color. And accumulation of carbohydrate, more amount of carbohydrate inside the plant may sometimes leads to less photosynthetic activity. And also the hormones. Next, here the factor affecting photosynthesis, like we have already studied our external and internal factors: light, water, temperature, wind speed and carbon dioxide can concentration of the carbon dioxide can reduce the photosynthesis. Based on this, Blackman has proposed a theory called law of limiting factor in 1905. What he said is, like, if there is a number of factors responsible for a particular reaction, then it will always depend to the factor that has the lowest means the lowest factor. Let's try to understand this with an example. Now, if you make a, you are asked to make a packet of mobile, iPhone mobile, where you have a mobile, you have got a charger, and you have got a packet. Now, say you have got 5 mobile, 7 charger, but only 2 packets. That means overall how many you will able to pack? You will able to pack only 2 mobile, because you only have 2 packets, where 2 mobiles will go and 2 charger will go. Rest of the thing will be waste because you don't have the packet to put them inside. That means here, which is the lowest? The packet was the lowest, and based on that only, the amount of number you could able to pack. Similarly here, if the particular factor is the lowest, say you have the lowest light, then here light will be the main responsible for the less photosynthesis, because you didn't have enough light to prepare photosynthesis, to carry out photosynthesis, even after you have lots of carbon dioxide and water in your environment. Here in the graph you can understand, like when you have a light, even if you got a higher light also, you will be in a stationary state. There is a particular area where the rate of photosynthesis is very higher. This is what you call the optimum light, or optimum uh, reaction, where at this particular point only, at a certain point of light intensity only, the photosynthesis rate will be higher. Similarly, if you have a carbon dioxide, even if you give, if we know that carbon dioxide is a requirement for photosynthesis, but if you give more amount of carbon dioxide, the plant will not carry higher rate of photosynthesis. At the particular optimum level only, it will carry very high rate of photosynthesis. Below that, it will again carry less. Above, it may have similar rate. Like if you can produce 10, like at less carbon dioxide you can produce only 5. At an optimum you can produce 10, but after getting more carbon dioxide than the optimum also, you cannot able to produce 12 glucose. Now also you can able to produce 10 glucose only. Similarly, as in case of temperature, I have already mentioned, higher temperature and lower temperature will badly affect photosynthesis. It will directly reduce if it is more less temperature, or if it is very high temperature. Only in optimum temperature you will able to get a good photosynthetic rate. Next, with the light we can say quantity, quality, and duration. Quantity I have already explained. Even if you have higher quantity also, the rate will not change. What is quality here? The quality is, as we explained, at 460, 470 something nanometer and 680 nanometer around, where it absorbs more light, and that is blue or the red light, and here the photosynthetic activity rate is higher. So if you give some like X-ray or UV rays, you will not get any photosynthetic activity. Your rate will not be higher. And duration is the time of exposure. So if you expose for 1 hour, the production will be less. If you expose for 7 hours, the production will be more, but the rate will be same. Example, if you produce 70 glucose at 1 hour, in 10 hours you will produce 700. You will not produce another 800. Means each and every hour similar rate only. Your rate of reaction will be same, but the amount will be reduced because you got only 1 hour to carry out your reaction. Carbon dioxide, 0.5% is considered as optimum, 0.3 to 0.4 is our atmospheric present, and 0.5% is considered as optimum. Above this, above 0.5% level of carbon dioxide may be harmful to the plant. In case of C3 plant, it usually takes 450 microliter of carbon dioxide. In case of C4 plant, it takes only 350 microliter of plant. I have already explained you, C3 plants are from the general plants that is present in a tropical region, where they will have lots of water, so more release of water during this process. They also release more carbon dioxide along with the water, hence they need to take more carbon dioxide for glucose production. Whereas C4 plant, they rarely open their leaves, and therefore only 350 microliter of carbon dioxide is required for them. Temperature, it mostly affect in the dark time. Temperature will affect mostly on the dark reaction. And water in the stress condition also, it will reduce the photosynthesis, because it will lead to the closure of the stromata in order to save itself from the release of carbon dioxide. from the release of carbon dioxide, and thus there will be no carbon dioxide and photosynthesis rate will be hampered. Leaf closure, if the leaves when the water get dry, the leaf will get curled and the size of the leaf will become small and light exposure will become less, and hence it will also reduce the photosynthetic activity. So I hope I am clear with this topic. Thank you very much for giving me this opportunity.